Welcome, dear viewers and professionals, for the current video on handling of out of specification. Commonly, it is called as OOS, and the pharma professionals who are working mainly in the industry are required to know this OOS, what is OOS, and also how to handle the OOS. OOS is out of specification. Whenever there is a result of the testing out of the specification, that means the OS is generated or the result is out of specification. Pharmaceutical industry involves manufacturing of the pharmaceutical formulations, testing of the pharmaceutical formulations in the laboratory. The pharmaceutical products are required to be safe and effective and should meet the quality requirements, and that is driven through the testing to meet certain criteria, or you can say to meet certain quality criteria. For example, potency or assay of the tablet formulation you can consider. The limit may be 90 to 110% of the label claim, or it may be 95 to 105% of the label claim. So 95 to 105% or 90% to 110% or 110% is the specification for assay. If the batch is tested for assay and if the result is not in this criteria or range of specification, then there is a OS or the result is out of specification. OS result is a critical aspect of maintaining product quality and the regulatory compliance in the pharmaceutical industry. So whenever OS is there, how to handle it is very important to understand. So this video is for handling of the out of specification results in the quality control labs. It is not likely that OS is because of the product problem, or it is only the manufacturing issue. It may be the testing issue. It may be because of the calculation error. It may be because of the documentation error. Human error may be there. Many times it is found that the OS is not actually the OS. It is just a human error to record or to follow the correct instructions for testing. So below is the structured approach for maintaining the OS, for managing the OS results, focusing on investigation, documentation, corrective actions, and ensuring regulatory compliance. Once the OS is there, the first initial assessment is to confirm the validity of the OS result, whether the result is itself correct or not. And the actions required for initial assessment is review the test result, check the clear errors in the data such as equipment malfunctions like calibration errors, weighing errors, sample contamination, human errors in the testings, human error in calculation, or wrongly putting the data. So if the equipment malfunction is there, then you cannot say that the batch is not okay or the product is not okay or product is failing in the test criteria. Cross-checking of the protocols is required to ensure the correct method is followed. An outcome of this initial assessment will be if the test result is found to be valid after reviewing, proceed with further investigation. So first is the checking the validity and confirming the validity of the OS result. If the result is not valid, then you cannot say that the result is in OS. But if the result is valid, that means OS happened and now investigation process should start. So first is informal investigation. Objective of the informal investigation is to conduct a preliminary investigation to identify any immediate assignable causes. So this is preliminary investigation. It requires certain actions like review the analytical records, including sample preparation, calibration, and method execution. We need to check whether the method is followed correctly or not. Check the environmental conditions at the time of testing, like temperature and humidity. Inspect the testing equipment to confirm was functioning correctly or not, and investigate staff adherence to SOP, that is standard operating procedures and the criteria, or the standard instructions. The outcome of this will depend on to the investigation happen. So if issue is identified, like example, incorrect sample handling or equipment malfunction. So if you have identified this, document the finding and proceed with retesting or corrective actions. If in the preliminary investigation it was found that the results are incorrect because of the incorrect sample handling or because equipment was not functioning properly, then these findings are to be documented, and next step is to proceed with retesting or corrective actions. Second is formal investigation. Initiate a formal and structured investigation if no assignable cause is identified during the informal phase. In some of the literature and documents, you may find that phase 1 and phase 2 investigations are there. So for this video, I am considering formal investigation when there is a no assignable cause identified during the informal phase or preliminary phase. Actions in formal investigations involve management and quality assurance personnel to oversee the investigation. Now you should know here that now the other departments like management and quality assurance people will take entry in the investigation process. Conduct thorough review of batch record to identify any manufacturing or process deviations that might have impacted the results. So whether it is only due to the testing or whether it is coming from the manufacturing of the batch. So now thorough investigation is required. Review raw data and analytical procedures to ensure compliance with the established protocols. Outcome of this will be to identify if there is an issue within manufacturing process or testing procedure. If no assignable cause is found, consider resampling and retesting. So this is very important to know when to go for resampling and retesting and when not to go for resampling and retesting, because in quality aspects meeting the criteria or having the compliance by resampling and retesting is not encouraged. So we should be known to these concepts. Third is documentation. Maintain comprehensive records to ensure traceability, compliance, and accountability. Document every step of investigation process, including initial test results, results of the informal and formal investigations, corrective action taken, including the equipment recalibration or SOP adjustment, and ensure compliance. Follow regulatory guidelines for documentation and ensure that all findings are reported accurately. Outcome will be a well-documented investigation is essential for both internal records and external audits, or regulatory inspections. Retesting protocol. The objective of retesting protocol is to confirm whether the OS result is accurate or caused by a procedural or sample related issue. If assignable cause is found, then invalidate the original OS result. If root cause, that is equipment failure, contamination, is identified, retest the sample according to established procedures, ensuring that the same sample set is used. Outcome of this will be, if retesting confirms compliance with specification, document the finding and release the batch if applicable. So if you found that the OS results are not correct and OS result or OS was generated or OS result was because of the equipment failure or sample handling problem or contamination, then retest the sample, and if it complies with the specification, then release the batch. And if no assignable cause is found, then actions will be like: consider resampling of the batch to perform new round of testing, retest the resampled batch in accordance with the standard protocol, and the outcome will be all retest results must meet specification for the batch to be deemed acceptable for the release. So first was retesting, second is resampling and retesting. If all the retest result must meet specifications for the batch to be deemed acceptable for release. Corrective actions. Address the root cause of the OS result to prevent recurrence. For example, if you found that because of the improper sampling or improper sample preparation, OS result was there, then what was the corrective actions taken? What will be the preventive action to prevent the recurrence? That means the same problem should not happen once again. Actions will be retrain the staff, whoever the analyst, whoever the people are working. Training should be given to them. If human error was a factor in OS result, retain the personnel or proper procedures and techniques. Recalibrate the equipment. Ensure that all retesting equipment is properly calibrated to maintain accuracy. Revise SOPs if necessary. Update standard operating procedures, SOPs, to reflect improvements in testing or manufacturing practices. Outcome: implementing corrective actions will prevent similar issues from occurring in the future and improve overall product quality. So this is the corrective action. Regulatory compliance. Ensure all actions taken in response to OS results meet regulatory standards. Whatever the investigation is performed, the actions are done, all should meet the regulatory requirements. Adhere to guidelines provided by the regulatory bodies like FDA, EMA, ICH, and other country-specific regulatory authorities. Ensure timely reporting of OS result to regulatory agencies if required. Document all actions in response to OS results, including investigations, corrective measures, and retesting results. Outcome: compliance with regulatory guidelines is essential for maintaining the integrity of the pharmaceutical company and avoiding legal and compliance issues. So OS is very critical. Review and release. Make an informed decision regarding the release of the batch. Actions: management review. Ensure that all OS results and investigations are reviewed by the management before making decisions regarding the release of batches. QA decision. Quality assurance personnel must verify that the batch meets all specifications before release. It is not like that if the batch was failing in assay, so it was corrected and the batch is released. It is not like that. The batch should meet all the specifications. Final documentation. All test results including OS from including the results of OS investigation must be finalized and documented. Only batches that comply with specification and have been thoroughly investigation done should be released for sale. Now we'll see the summary of key steps in handling the OS. Initial assessment. Review the validity of the OS result. In this stage, check whether the OS result is correct or not. Investigation process. Informal and formal investigations to identify assignable causes. Documentation. Keep detailed record and records for regulatory compliance. Retesting protocol. Perform retesting if assignable causes are identified or resample if needed. Corrective actions. Implement corrective actions to prevent recurrence. Regulatory compliance. Ensure all actions comply with regulatory guidelines. Review and release. Review OS findings before deciding on batch release. So this is the information on handling out of specification results. In my experience, I come across a situation where the OS happened and the OS was closed based on the human error for making the sample for testing. Training was done and then the OS was closed. But when the regulatory authority inspected and audited the company or organization, that time they found that many of the OS are closed based on the human error only or analyst error only, and there is no proper justification, there is no proper corrective action taken. So the regulatory authority has raised many queries on the OS handling. Whenever you are working in the regulatory environment in the pharmaceutical industry, the OS result handling or OS procedures and the batch release is very critical, and it is very important to know the OS handling. So I hope this video will help you to have some understanding about handling of the OS results. Thank you for watching the video, and if you have any questions, please comment. Thank you.